Good evening students. Today I am going to discuss about the computational modeling of drug disposition from the chapter 2 of CAT. Here we have introduction. Drug discovery has focused almost exclusively on efficacy and selectivity against the biological target. As a result, nearly half of drug candidates fail at phase 2 and phase 3 clinical trials because of undesirable drug pharmacokinetic properties. The pressure to control the escalating cost of new drug development has changed the paradigm since the mid 1990s. To reduce the attrition rate at more expensive later stages, in vitro evaluation of ADMET properties in the early stage of drug discovery has been widely adopted. Many high throughput in vitro ADMET property screening assays have been developed and applied successfully. For example, CACO2 and MDCK cell monolayers are widely used to simulate membrane permeability as an in vitro estimation of in vivo absorption. These in vitro results have enabled the training of in silico models, which could be applied to predict the ADMET properties of compounds. Fueled by the ever-increasing computational power and significant advances in silico modeling algorithms, numerous computational programs that aim at modeling drug ADMET properties have emerged. A comprehensive list of available commercial ADMET modeling software has been provided previously by Van de Waterbeemed and Gifford. Modeling techniques. There are two main approaches of the drug modeling techniques: quantitative approach, qualitative approach. Quantitative approach, represented by pharmacophore modeling and flexible docking studies, investigate the structural requirements for the interaction between drugs and the targets that are involved in ADMET process. Especially useful when there is an accumulation of knowledge against a certain target. For example, a set of drugs known to be transported by transporter would enable a pharmacophore study to elucidate the minimum required structural features for transport. Three widely used automated pharmacophore perception tools are DISCO, distance comparisons, GASP, genetic algorithm similarity program, Catalyst or HipHop. The application of different flexible docking algorithms in drug discovery has recently been reviewed. The essential interactions derived from either study can be used as screen in evaluating drug ADMET properties. Qualitative approaches, represented by quantitative structure activity relationship and quantitative structure property relationship, utilize multivariate analysis to correlate molecular descriptors with ADMET related properties. A diverse range of molecular descriptors can be calculated based on the drug structure. It is important to select the molecular descriptors that represent the type of interactions contributing to the targeted biological property. It is essential to select the right mathematical tool for most effective ADMET modeling. Sometimes it is possible to apply multiple statistical methods and compare the result to identify the best approach. A wide selection of statistical algorithms is available to researchers for correlating field descriptors with ADMET properties, including simple multiple linear regression, multivariate partial least squares, and the nonlinear regression type algorithms, such as artificial neural networks and support vector machine. These are the qualitative approaches. Next one, drug absorption. Because of its convenience and good patient compliance, oral administration is the most preferred drug delivery form. As a result, much of the attention of in silico approaches is focused on modeling drug oral absorption, which mainly occurs in the human intestine. Drug bioavailability and absorption is the result of the interplay between drug solubility and intestinal permeability. First one, solubility. A drug generally must dissolve before it can be absorbed from the intestinal lumen. By measuring a drug's log P value and its melting point, one could indirectly estimate solubility using the general solubility equation. To predict the solubility of the compound even before synthesizing it, in silico modeling can be implemented. There are two main approaches to modeling solubility based on the underlying physiological process. Empirical approach. The dissolution process involves the breaking up of the solid from its critical lattice and the association of the solute with solvent molecules. Empirical approaches represented by QSPR utilize multivariate analysis to identify correlations between molecular descriptors and solubility. Even though the calculation process ignores the underlying physiological process, the molecular descriptor selection and model interpretation still requires understanding the dissolution process. Selection of field descriptors that adequately describe the physiological process and the appropriate multivariate analysis is essential to successful modeling. The target property for most models is the logarithm of solubility, and many models are trained and verified with the Aquazole and FISPROP databases. Next one, intestinal permeation. Intestinal permeation describes the ability of drugs to cross the intestinal mucosa, separating the gut lumen from the portal circulation. It is an essential process for drugs to pass the intestinal membrane before entering the systemic circulation to reach their target site of action. The process involves both passive diffusion and active transport. It is a complex process that is difficult to predict solely based on molecular mechanism. Most current models aim to simulate in vitro membrane permeation of CACO2, MDCK or PAMPA, which have been useful indicator for in vivo drug absorption. To correctly predict overall oral absorption, drug metabolism in intestinal epithelial cells by cytochrome P450 enzyme should also be considered. Other than the different approaches mentioned above, commercial packages such as GastroPlus, Simulations Plus, Lancaster CA and IDEA and Line Bioscience in Cambridge MA are available to predict oral absorption and other pharmacokinetic properties. They are both based on the advanced compartmental absorption and transit CAT model, which incorporates the effects of drug moving through the gastrointestinal tract and its absorption into each compartment at the same time. Next one, drug distribution. Distribution is an important aspect of drug's pharmacokinetic profile. The structure, physiochemical properties of a drug determine the extent of its distribution, which is mainly affected by three parameters: volume of distribution (VD), plasma protein binding (PPB), blood-brain barrier (BBB). First one, volume of distribution. VD is a measure of relative partitioning of a drug between plasma and tissue. An important proportional constant that, when combined with drug clearance, could be used to predict drug half-life. The half-life of a drug is a major determinant of how often the drug should be administered. However, because of the scarcity of in vivo data and the complexity of the underlying process, computational models that are capable of predicting VD based solely on computed descriptors are still under development. Next one, plasma protein binding. Drugs bind to a variety of plasma proteins such as serum albumin, as unbound drug primarily contributes to pharmacological efficacy. Plasma protein binding is an important consideration when evaluating the effect to drug plasma concentration. The models proposed on PPB should not rely on the binding of the only one protein, while plasma protein binding, because it is a composite parameter reflecting interaction with multiple proteins. Third one, blood-brain barrier. The BBB maintains restricted extracellular environment in the central nervous system. The evaluation of drug penetration through BBB is an integral part of the drug discovery and development process. Again, because of few experimental data derived from the inconsistent protocols, most BBB permeation prediction models are of limited practical use despite invasive efforts. Most approaches model log blood or brain log BB, which is measurement of the drug partitioning between blood and brain tissue. The measurement is an indirect implication of the BBB permeability, which does not discriminate between free and plasma protein bound solute. Next one, drug excretion. Drug excretion or clearance of a drug is quantified by plasma clearance, which is defined as plasma volume that has been cleared completely free of drug per unit of time. Together with VD, it can assist in the calculation of drug half-life, thus determining dosage reason. Hepatic and renal clearances are the two main components of plasma clearance. No model has been reported that is capable of predicting plasma clearance solely from computed drug structures. Current modeling efforts are mainly focused on estimating in vivo clearance from in vitro data. Just like other pharmacokinetic aspects, the hepatic and renal clearance process is also complicated by the presence of active transporters. Now, active transporters. Active transporters should be an integral part of ADMET modeling program because of their ubiquitous presence on barrier membranes and the substantial overlap between their substrates and many drugs. Unfortunately, because of our limited understanding of transporters, most prediction programs do not have a mechanism to incorporate the effect of active transport. However, interest in these transporters has resulted in a large amount of in vitro data, which in turn have enabled the generation of pharmacophore and QSAR models for many of them. These models have assisted in the understanding of the complex effects of transporters on drug disposition, including absorption, distribution, and excretion. Their incorporation into current modeling programs would also result in more accurate prediction of drug disposition behavior. Active transporters, one of the transporter PGP, P-glycoprotein, is an ATP-dependent efflux transporter that transports broad range of substrates out of the cell, affects drug disposition by reducing absorption and enhance renal and hepatic excretion, and also responsible for multi-drug resistance in cancer chemotherapy. Because of its significance in drug disposition and effective cancer treatment, PGP attracted numerous efforts, has become the most extensively studied transporter with abundant experimental data. By comparing and matching all GP pharmacophore models, common areas of identical chemical features such as hydrophobes, hydrogen bond acceptors, and ring aromatic features, as well as their geometric arrangement, were identified to be the substrate requirements for PGP. Next, BCRP transporter, breast cancer resistance protein, is another ATP dependent efflux transporter, confers resistance to a variety of anti-cancer agents including anthracyclines and mitoxantrone. In addition to high level of expression in hematological malignancies and solid tumors, BCRP is also expressed in intestine, liver, and brain, thus implicating its intricate role in drug disposition behavior. This model emphasizes very specific structural feature requirements for BCRP, such as the presence of 2,3 double bond in ring C and hydroxylation at position 5. Satisfying the transport model would render a compound susceptible to BCRF, but not fitting into the model does not necessarily exclude the candidate from BCRP transport. Next, third one, nucleoside transporter. Nucleoside transporters transport both naturally occurring nucleosides and synthetic nucleoside analogs that are used as anti-cancer drugs, example cladribine, and antiviral drugs. Different types of nucleoside transporters are concentrative nucleoside transporters CNT1, T2, T3, equilibrative nucleoside transporters ENT1 and T2. Each have different substrate specificities. The broad affinity, low selective ENTs are located. The high affinity selective CNTs are mainly located in epithelium of intestine, kidney, liver, and brain, indicating their involvement in drug absorption, distribution, and excretion. A more comprehensive study generated distinctive models for CNT1, CNT2, and ENT1 with both pharmacophore and 3D QSAR modeling techniques. Next, fourth one, HPEPT1, human peptide transporter. Low affinity, high capacity oligopeptide transport system that transports a diverse range of substrates including beta lactam antibiotics and angiotensin converting enzyme inhibitors, mainly expressed in intestine and kidney, affecting drug absorption and excretion. A pharmacophore model based on three high affinity substrates, glycer, bistatin, and enalapril, recognized two hydrophobic features: one hydrogen bond donor, one hydrogen bond acceptor, one negative ionizable feature to be HPEPT1 transport requirements. Fifth one, ASPT, apical sodium dependent bile acid transporter, a high efficacy, high capacity transporter expressed on the apical membrane of intestinal epithelial cells and cholangiocytes. It assists absorption of bile acids and their analogs, thus providing an additional intestinal target for improving drug absorption. The model revealed ASPT transport requirements as one hydrogen bond donor, one hydrogen bond acceptor, one negative charge, and three hydrophobic centers. And sixth one, organic cation transporters facilitate the uptake of many cationic drugs across different barriers, membranes from kidney, liver, and intestine epithelia. Antiarrhythmics, beta adrenoreceptor blocking agents, antihistamines, antiviral agents, and skeletal muscle relaxants. These OCTs have been cloned from different species, OCT1, OCT2, and OCT3. The model suggests the transport requirements of human OCT1 as three hydrophobic features and one positive ionizable feature. Next, seventh one, OATP, organic anion transporting polypeptides, influence the plasma concentration of many drugs by actively transporting them across a diverse range of tissue membranes such as liver, intestine, lung, and brain. Because of their broad substrate specificity, OATPs transport not only organic cationic drugs as originally thought but also organic cationic drugs. Currently, 11 human OATPs have been identified, and the substrate binding requirements of the best studied OATP1 B1 were successfully modeling with the metapharmacophore approach recently. And the last, BBB, choline transporter, native nutrient transporter that transports choline, a charged cation, across the BBB into the CNS. Its active transport assists the BBB penetration of choline-like compounds, and understanding its structural requirements should afford to more accuracy prediction of BBB permeation. Three hydrophobic interactions and one hydrogen bond interaction surrounding the positively charged ammonium moiety were identified to be the important for BBB choline transport recognition. Thank you.